So last but not least, before the break, our next speaker is Professor Cyrille Cohen from Ilan University, who heads the immunology lab. Cyrille became a celeb during this pandemic. He is a member of the Advisory Committee for Clinical Trials on SARS-CoV-2 vaccines at the Ministry of Health in Israel. But today you will speak about immuno-oncology, right? Thank you.
Thank you so much, Ronit. I have to tell you, I'm very happy to be here in a face-to-face meeting. Hasn't happened for 2 years. It's okay, I'm going to take care of that, don't worry. And I still don't know why. Still asking myself, they're talking about a virus in the news, but I don't believe them. Okay, so basically, what do we want to talk about today? So what I would like to do is actually talk about a new way to potentiate the immune system and to improve cell therapy. And basically, as we just heard, T cells are very important. We are really focusing on T cells in my lab. And T cells need two main signals in order to get activated. One is what we call the TCR signal to recognize the antigen. The second signal is what we call a co-stimulatory signal to boost that response and enhance proliferation, survival, and activation. And there are a lot of co-stimulatory molecules and also co-inhibitory molecules that are trying to balance the immune system. The question is really, why do we need to have some co-inhibitory molecules? So one of the, I would say, the reasons why that it can prevent autoimmune diseases. But we published a work together with colleagues in which we showed that it's not only to prevent autoimmune diseases, but it's also to prevent T cells from burning out when they are responding. So in the course of their life, T cells will get the opportunity to interact with several cells, and therefore there will be several interactions between, do we see that? Yeah, glowing. So, yeah. So T cells will get the opportunity to, okay. I'm gonna do that with this. Okay, so T cells will get the opportunity to interact through different receptors. And based on that, based on this kind of activation, we know that there are three main therapies that are based on T cells against cancer. One that I won't talk about, and we've been involved also in this, is TIL therapy, tumor-infiltrating lymphocyte therapy. And the second one, or second type, is more genetically engineered T cell therapy that can be done through either the expression of a CAR, a chimeric antigen receptor, Zeli Geschrar and Gidi Gross were the pioneers in that work, of course, or TCR, which has been, I would say, in the making for the past 15 years. I have the privilege to be in the group that developed that therapy 15 years ago. And basically, the idea is to take a T cell and to introduce a receptor, either a native like the TCR or an engineered receptor, so that the T cell will recognize a cancer cell. But there is a very subtle difference between those two, I would say, therapies. While there is, antigenic I would say antigen recognition moieties in both cases. Here it's an antibody, usually, not always. I'll show you at the end of my talk that we are developing therapies that are not based only on antibodies, but also on natural receptors. While there is an antigen recognition moiety, there is also an activation inside the cell moiety, but here in the CAR, usually you have also a co-stimulatory part, and you are lacking that in TCR. So how can we bring co-stimulation to T cells? And one of the things that we have noticed in the course of our, I would say, studies is that the primary structure of these receptors, chimeric receptors, is extremely important for, I would say, the therapy. Here I'm just showing a few results in a collaboration we have with Adassa, with Polina Stepensky and her group, Shlomit and Natalie, in which we developed different CARs, chimeric antigen receptors, against multiple myeloma. And just to make a long story short, even if you get all those receptors expressed, at the end of the day, in mouse, you see, and now in patients, thanks to this collaboration, you see that really a few amino acids difference, for example, in the linker region, can make a great difference in vivo. And we've been trying to understand those differences so that when we look at those receptors, I would say the way we build those receptors, we understand that primary structure is very, very important. So again, as I told you, CAR are very prominent, very, I would say, as a therapy, very promising in hematological malignancies, but we are still lacking some, I would say, efficient CAR therapies in solid tumors. There are different, I would say, reasons for that. But I have to tell you that TCRs as a TCR T-cell as a therapy has a great advantage because it can target much more antigen than, I would say, CAR T-cells. And still, as I told you, we are lacking that co-stimulation. So what can we do to bring about or to bring to those T-cells with a TCR some better co-stimulation signals? So that's more or less the idea we had a few years back. We know that the ligands for co-stimulatory molecules are usually not expressed by cancer cells. Okay, there's no reason for that. Cancer cells don't want to overstimulate the immune system, at least, T cells. What they do express are ligands that can activate co-inhibitory molecules in T cells, meaning that even if there is a recognition through the TCR, no matter now if it is engineered or natural TCR, this pathway can actually shut down the function of T cells. And we try to think how we can circumvent that, especially when we are talking about a T cell that we are introducing a new TCR specific for a cancer antigen. And we came up with that idea. We actually designed a chimeric receptor that is on the outside able to bind PD-L1 that is overexpressed by cancer cells, but on the inside, the endodomain will be actually derived from a co-stimulatory molecule. And that chimeric molecule that we call PD-1 CD28, that was the first co-stimulatory switch receptors we developed, was actually able to, I would say, slow down the growth of tumors, was able to sustain proliferation inside the tumor of T cells that express that molecule but not a control molecule. So for us, this was a very important step in order to prove that you can add to TCRs, in that case a TCR specific for melanoma, you can add another molecule that will help those T cells to perform their job. Problem is that PD-L1, the ligand of PD-1, is not always expressed at tumor sites, and therefore we said, okay, let's go and try to develop this in a more generic way and target different co-inhibitory receptors or use different co-inhibitory receptors in order to have a panel of potent CSRs, co-stimulatory switch receptors. So there are, as I told you, a lot of receptors, and we did some work on the TIGIT molecule, the TIGIT pathway, which is also very important in T cells, and the activation by CD155, which is one of the ligands of TIGIT, so the ligation of CD155 on TIGIT that is expressed by T cells can actually dampen the T cell function. So we said, okay, let's develop a new receptor that will be based on the extracellular domain of TIGIT and the intracellular domain of CD28. First, we made sure that tumors can express, in that case melanoma, can express high levels of CD155. Second, we built different receptors, actually. We built two receptors with different transmembrane, and also one receptor that had no intracellular domain as a control to show that the intracellular domain of CD28 is essential for the function of those co-stimulatory switch receptors. So all those receptors were actually were able to be expressed in T cells. And what we saw actually, and that was what we expected, is that when you take T cells just with a TCR, just with a signaling molecule that recognizes an antigen like this molecule, the F4 molecule, and you co-culture that with a melanoma cell, so you get secretion, for example, of interferon gamma or TNF alpha at what we call, 100. We normalize our results to that. When you add a co-stimulatory switch receptor with CD28 intracellular domain, you can boost that cytokine secretion. If you put a chimeric receptor without the CD28 intracellular domain, we call that stop or TIGIT stop, what you could see is that you lose that effect. So the intracellular molecule or a portion of that molecule is important for the function of co-stimulatory switch receptors, and we decided to focus on one of those two, the one with the native transmembrane domain of TIGIT. We were able to show that when you co-culture T cells equipped with a TIGIT CD28 molecule, you get better activation. You get also a better signaling in terms of phospho-ERK compared to T cells that expressed only the TCR. And also better, I would say, prevention of apoptosis with the expression of BCL-XL in those engineered T cells. Now, we know that T cells can be divided into CD8 and CD4 T cells. CD8 T cells usually recognize MHC class 1, and I say usually, and CD4 usually recognize MHC class 2. We have actually, a few years back, we have described the occurrence of a population of, this is for the immunologists here, are there. We have described a population of CD4 T cells that recognize MHC class 1, by the way, in patients. But in any case, we wanted, okay, here it is. We wanted to show that this molecule, the TIGIT CD28, can perform both in CD8 and CD4 T cells. So we sorted those cells, co-cultured them with different tumors, and as you can see, adding this TIGIT CD28 can really boost the response of T cells against different tumors, whether they are CD8 or CD4, which is for us very important. We also wanted to mimic the conditions that T cells encounter in the tumor. Basically, most of, I would say, the problems we encounter with T cells is the exhaustion state, in which T cells are repeatedly exposed to the same antigen inside the tumor, and there is also the activations of PD-1 pathway and other pathways I told you. And we say, can we mimic that in the lab? Not only, of course, in animals. So what we did was to develop an original assay in which we would generate engineered T cells. We will put them with tumors, and every few days we will replenish new tumor cells, meaning that we will keep on, I would say, stimulating those T cells for at least 8 days. And what we did was to divide that in T cells that were engineered to express the TIGIT CD28 molecule or native T cells. What we could see is that when you expose T cells for 8 days to an antigen-positive target, it will induce some exhaustion, okay, compared, for example, to an antigen-negative target. So you see there is a drop in, for example, interferon gamma secretion. But when you add actually that TIGIT CD28, you can actually remedy to that and prevent this kind of hypofunctional assay, meaning that for us it is very relevant to introduce that molecule in T cells that are going to fight especially solid tumors and that are exposed to, I would say, immunosuppression. So we took that also to a mouse model of human tumors, a xenograft model, and here, as you can see, the TIGIT CD28 molecule was able to slow down the growth of tumors and lead to a better survival of mice that were injected with T cells expressing TIGIT CD28. Point is that, as I told you, even TIGIT or the ligand for TIGIT, which is CD155, is expressed only by certain tumors. And we wanted a more general way to tackle tumors and to bring, I would say, that concept of added co-stimulation to target different types of tumor. So we focused actually on glycosylation and on receptors in T cells, and not only in T cells, also in NK cells, that are able to recognize glycosylated residues on tumor cells. So there is a big family of those receptors that are expressed by white blood cells that are called Siglec. And I'm not going to go into what each receptor is doing. Suffice to say that we focus on Siglec-9, the Siglec-9 receptors that we believe is a new checkpoint for T cells, and we have a work. I'm going to show you some unpublished results that we are about actually to publish, hopefully. Basically, we decided to focus on Siglec-9 that is expressed. It was known to be expressed by NK cells, but not by T cells. So we worked on that molecule, and we worked also on the ligands. So now the ligands are actually glycosylated molecules. We have ways to measure the level of those ligands on different tumors. What we could see, which was extremely interesting, is that, like, for example, PD-L1, that is, I would say, overexpressed following incubation with, overexpressed by tumor cells that are incubated with interferon gamma, we could also show that if you take tumors and incubate them with interferon gamma, they will upregulate that glycosylation pattern on the surface that might lead to immunosuppression. We have also the mechanism. We show that also on the level of the enzymes that are able to do that, but I don't have time to show you. On the other side, meaning the side of the T cells, what is happening? We also find a very interesting, I would say, phenomenon in which we studied around 20 different donors, most of them healthy donors. And what we could see is that following activation, after a few days of cultures, there were half of those donors that were overexpressing the Siglec-9 molecule, and half of them, very low level. Each of them, went, in each of the donors, T-cell donors, you could see, an upregulation of the Siglec-9 receptor over time. But there were striking donors that were, we call them Siglec-high donor. So we have done some, and I don't have the time to show you, but we have done some RNA-seq on those different populations to try and understand, what are the tenets of this kind of phenomenon. But suffice to say that this is very interesting. The other thing we did in collaboration with Ilan was to knock down the Siglec-9 receptor in high donors. And interestingly enough, when you knock down that receptor and co-culture T cells with different tumors, you can see a better cytokine secretion, a better activity compared to the same cells that were not CRISPRed out or knocked out for Siglec-9. And now to, again, link to what I was telling you a few minutes before, and these are my last slides, what we said is that, okay, Siglec-9 is actually a checkpoint, and if it is a checkpoint, perhaps we can act on it and use it as a backbone to develop also a chimeric switch receptor that, when introduced in a T cell that has a specific TCR against an antigen, that molecule, which is Siglec-9 on the outside, but in the inside, as an endodomain that is co-stimulatory, and we try different co-stimulatory molecules. This will help in a broader way, because we have noticed, and I can't go into details, but I showed you that there is an upregulation of glycosylation in a different set of tumors, very different tumors. So this is actually promising for us, because it might be a universal target for us, this kind of glycosylation. So we can help T cells with a TCR to get a better activity. So we did that. We developed different chimeras. Here again, the primary structure of those receptors was extremely important. Even a few amino acids here can make a huge difference in the function of those new receptors. And actually, we like that in my lab, playing around. It's like Lego. I always liked Lego when I was a kid. Playing around with a few sequences and show dramatic differences. I'm just going to show you my last slide. Actually, I have 2 slides. And say that when you take mouse that are inoculated with a human tumor. When you do nothing, when you inject nothing, of course, those mice are dying. When you inject T cells that express a specific TCR, this can delay the growth of the tumor. But when you inject T cells that have a knockout of the S9, Siglec-9 receptor, it can, to some extent also slow the development of the tumor. But the best results was to have a T cell that expressed a TCR that was CRISPRed or to abrogate the native expression of Siglec-9, and also that express a co-stimulatory switch receptor. And as you can see, those mice are alive and well, actually. And just again to refer to what I just said before, when you use different target sequences in those receptors and you build your switch receptors a little bit differently, you can see a real difference in the function. These are receptors with almost the same elements. It's just the way you put them and organize them in the molecule. So, just on time. Basically, you can improve genetically T-cell therapy. We need better T-cell therapies. CAR T-cells are excellent. Again, we are very fortunate to participate in that effort clinically, but again, we know the limitation of CAR T-cell therapy. We need better TCR therapies, and what we believe is that beyond the fact that you can improve the TCRs, you can also improve T-cells expressing TCRs through the expression of chimeric switch receptors. I want to thank all my collaborators, especially Vasily and Shiran, that have been working on the last bit of the project that I told you. Our collaborators in Hadassah Medical Center, Professor Michalotem, with whom we have also an upcoming clinical trial based on a molecule we developed, and Professor Polina Stepensky, our friends in different universities here and abroad, and also Aya Lendel in Bar-Ilan University. And thank you very much for your attention. These are the people making the work. Thank you so much.
Wow, so many hands raised. Thank you, Cyrille.
No, thank you. Being naked without my mask.
Can you hear me? Okay, thank you, Cyrille. This was fascinating. So many questions. Dino, start.
Really?
So first, I have several questions, but I leave them for the break. So one main that maybe I missed, do you need to also disrupt the endogenous inhibitory signal to introduce you switch? Because you didn't say anything about that. If you are disrupting the, let's say, TIGIT, the normal one, because it will be a kind of a counterbalance, right, from the cells that will try to, like you mentioned, there are other inhibitory, and they will maybe try to overexpress the others. So maybe I missed that you have to also knock down in terms of introducing your switch.
Yeah, so
One that we get a lot for CAR T, and I'm asking here, if you introduce this switch, what about autoimmunity, right? The autoimmunity of the ones that have the TCR endogenous.
okay, so these are great questions. So basically, in most of the works we've been doing so far with PD-1 and TIGIT, we didn't need, or we didn't knock down the original receptor, neutralize the signal from the original receptor, and we got some effects. The question is really, what could we have done to get a better signal? But that's what we did actually here, and that's what you're seeing here. These are regular T cells, These are T cells engineered with the switch receptor and the TCR, but these are T cells engineered with the switch receptor, but also knocked down for the original receptor, and you can see that it seems much better. So yes. And the second question regarding autoimmunity, the point is that, and this is a great question, the point is that we are, in all the experiments I showed you, we are co-expressing a TCR. Okay, this is an example. We are co-expressing a TCR that is specific for a tumor antigen with this switch receptor. I haven't showed you that, but basically if you just express the core receptor or the switch receptor alone, you don't get activation. So that's, I would say, for us, a way to ensure to some extent that we will not promote autoimmune manifestations.
Hi, great talk.
Awesome.
Have you tried coupling the natural competitors? For example, TIGIT with CD226, because they compete for CD155, or CD28 with CTLA4.
CTLA4 and, for example,
Exactly.
CD28, etc., etc. So,
Just because this in this competition, the signal that is sent from TIGIT is much stronger than CD226, for example.
yes, you are right. So we play so CD226 is DNAM. So we played a little bit with that. But to tell you the truth, so far the best results we got were either with CD28 or with 41BB. Actually, 41BB in the last, in the last bit of data I showed you was the strongest in, I would say, intracellular domain. We are trying to play a little bit with third generation, trying different signal in the same molecule, but this is not easy, I have to tell you, because proteins sometimes have a funny way to fold inside the cell, so we have also to find the right linkers between all those, I would say, in endo domains. But again, that's a great question. Thank you.
Yes, hi Cyrille. Did you look in the tumors when you kill the mice where these T cells are present and whether you see differential localization, specifically because of the glycosylation signal, which could be affected by microenvironment and so?
Yeah, so we haven't done that. In the past, we developed together with Rachela Popovtzer ways to locate, I would say, T cells, to follow on T cells, on the migration of T cells with gold nanoparticles, but we are planning to do some immunohistochemistry. We need a good pathologist, if you know one, tell us, so that we can actually better look at the interaction, in situ. But this is also a great question.
Can you hear me now?
Yes.
One more question regarding the models. You were showing models of human cancers in immunodeficient mice, I assume.
Yes.
So what is the contribution of the resident T cells or any other counterparts? There was no...
We are using NSG mice. You have no T cells there. So we don't believe there is an effect. We do believe, by the way, that, and we're working on that, that when you introduce human T cells in a mouse and you are trying to look at the development of tumors, sometimes we see a funny thing that just T cells, I think that this is because they are activated, but just T cells can promote tumor growth. So we are looking into that. So what will be the influence of resident T cells? I think we'll know that only in patients.
In other populations, like in the example of PD-L1, we know that is also expressed on dendritic cells. So.
Yeah, that's correct. That's correct. So for that, you need a better. So we've been playing around with that idea of taking NSG mice, introducing stem cells, getting exactly. So I'll tell you more when we do the experiments.
Thank you, Cyrille. Thank you
very
so
much.
much for the first part of this wonderful symposium. We have 20 minutes break, so we'll come back at 11:20 Israel time. It's still okay in terms of time because unfortunately, Professor Gidi Rehavi will not be able to join us, also sick at this time. So we'll be back at 11:20. Thank you. We wish them all well.